just to warm up the audience. But hello, my name is Cheyenne and today I'm going to be talking to you about the manifestations of systemic health and disease in the oral microbiome. So before we really get into it, first I just want to go over what exactly are microbes. So microbes are mostly prokaryotic cells. Our bodies are made up of eukaryotic cells, which as you can see here are around 10 to 100 micrometers large. To put that into perspective, 1 millimeter is around 1,000 micrometers. So these are incredibly small cells. And the prokaryotic cells that make up microbes are about 0.25 to 10 micrometers large. And they also outnumber our cells in our body. There are about 10 prokaryotic cells for every eukaryotic human cell. So essentially there are very, very tiny cells and we have a lot of them in our body. Now what exactly is a microbiome? A microbiome is a collection of these microbes, and within that collection the microbes create symbiotic relationships with each other. A symbiotic relationship is essentially where the byproduct of one microbe is used as the nutrient of another microbe and back and forth, vice versa. A good metaphor for this relationship is the way we interact with plants. So plants release oxygen, which we then breathe in, and then we exhale carbon dioxide, which the plants then use for their own respiration and metabolism. And how exactly do these microbiomes form? Well, everything we do in our life, everything we touch, everything we eat, everything we drink contains microbes. And so as we go throughout our life and touch and eat various things, we absorb and, not necessarily absorb, but we take those microbes and they grow and aggregate in our body and create microbiomes throughout our body. Microbiomes are also very unique based from person to person because everybody has their own lifestyle and genetics, etc. So here is a agar plate where a person put their hand on agar gel, which transferred the microbes from their hand onto the gel, and they allowed the gel to grow. And as you can see, there's a huge diversity in the microbes that grew on that gel. And if you compared this gel to the handprint of another person, it would look very different. So people's microbiomes are very unique. However, the specific type of environment that a microbiome is in is a better indicator of the types of microbes that will grow in that microbiome than the individual themselves. So what I'm trying to get at here is that the oral microbiome of one person is going to be more similar to the oral microbiome of another person than to their own gut microbiome, because the oral cavities of two people are going to have more similar environments than the gut and the oral cavity. Now, how are microbiomes important to our health? This is actually a relatively new idea in the scientific field. Around 15 years ago, there was a study done about obesity where they transferred the gut microbiota from lean patients and obese patients, and they transferred them into germ-free mice. Germ-free mice are basically mice that haven't had anything done to them yet. And then they allowed these mice to grow, and they found that the mice that received the microbiota from lean people either stayed or became lean, and then those who received the microbiota from the obese people became obese. So from this study, we learned that the microbiome is very important in determining our health. And using this study as a jumping off point, more studies were done to find what factors affect our microbiome. And they found that eating fermented foods was a huge factor, because fermented foods contain microbes that create byproducts that are very beneficial for our health. Also, eating a very diverse and high-fiber diet was shown to improve the microbiome of people, particularly in the gut, because the diversity of the diversity of one's diet directly correlated with the diversity of their microbiome and fostered better byproducts from that microbiome. All of these things, all of these research studies were mostly focused on the gut microbiome, and that's why they have to do with a lot of diet-related things. But I would like to point out that all food and microbes go through the mouth first. So why aren't we talking about the oral microbiome? Well, we're about to, so don't worry. The oral microbiome, although it is a small cavity, there are several microbiomes within the oral cavity, and there can be differences in the types of microbes that grow based on the specific environment within your oral cavity. So for example, the hard palate has a lot of streptococcus and lactobacillus. The tongue has a lot of streptococcus, actinomyces, and fusobacterium. The saliva has a lot of streptococcus. The tonsils have a lot of streptococcus and fusobacterium. The throat has a lot of actinomyces and fusobacterium, whereas the buccal mucosa has a lot of streptococcus. So of course these aren't the only bacteria in the mouth. There have actually been over 700 species of bacteria identified in the oral cavity alone. But this is just to give you an idea that although there are some similarities between the microenvironments in the mouth, like Streptococcus is pretty much everywhere, there are subtle differences such as lactobacillus only being on the hard palate, and those subtle differences are because of the subtle differences in the environments like oxygen content and specific nutrient content. Zooming in on something you're probably a bit more familiar with, the tooth and the gum line. So here, obviously on the tooth, there's a lot of access to oxygen, so the tooth is going to be primarily occupied by aerobic bacteria, and then the byproducts produced by these bacteria are going to cause things like dental caries or cavities. And then on the gum line, there is much less access to oxygen, so the bacteria that populate that area are then going to be primarily anaerobic bacteria. Now, we know that these microbes populate our mouth, and we know that they eventually cause things like dental caries and periodontal disease. And we know that in order to prevent those things, we have to brush and floss our teeth. But what is brushing and flossing really doing? Well, there are these things called biofilms, which are essentially layers of bacteria that, as they aggregate over time, they begin to create symbiotic relationships with each other. And they grow and grow more and more. And once they get to a high enough concentration, they begin releasing these things called virulence factors, which are essentially compounds that make their surrounding environment more favorable to their own survival, and they can reproduce even more. And once enough of these virulence factors accumulate around the bacterial layer, it creates a protective layer, not only because it fosters the optimum growth of the bacteria themselves, but it also creates a protective layer from possible invaders, such as pathogenic bacteria and antibiotics. And how do these biofilms form in the oral cavity? Well, first, there is the initial attachment of glycoproteins from our saliva. So this happens within seconds after brushing our teeth, whenever we lick our teeth or close our mouth. Once the glycoproteins attach, bacteria from the rest of our body, or if we eat something or drink something, bacteria from those external sources attach to the glycoproteins, and then they aggregate and reproduce until they are in high enough concentration that they create that protective layer around them. Now this is where we would officially call it a biofilm, but if the biofilm is not disturbed at this point by like brushing or flossing your teeth, then they continue to overgrow, and then the bacteria becomes in such high concentration that it essentially bursts out of the biofilm and can either attach elsewhere in the mouth or travel elsewhere in the body. And that's going to be a key point there. Now, what populates these biofilms? That is dependent on a lot of things, which is why they're so different from person to person. But going through the factors, first there is the host lifestyle. The host is just the person whose oral cavity we're looking at. There's the host lifestyle. So this will be things like diet mostly, whether they have a high sugar versus a high fiber diet. That's going to affect the types of microbes that grow in the oral cavity. The next is the host genotype. So if someone has a genotype that where they don't produce as much saliva, that's going to be a big factor. The immune system. So based on a person's immune response to the types of bacteria that grow in their mouth, that can affect both the type and the amount of bacteria that can grow in their mouth. The host environment, so a big thing here is whether or not the person drinks fluoridated water, which most of us do, but some people do not. That can really affect the amount of bacteria that can grow in the mouth. And then the host's physiology. This is mostly age related, so things like blood flow and the type of diet that the person is able to eat is very dependent on the host's physiology or age. And you see that I've written core oral microbiome right here. And this is something that we're very interested in if we want to use the oral microbiome as a means of identifying disease. So before we get into why it's important, let's talk about how we're going to find it. So the way we find a core healthy oral microbiome is we first analyze the microbiomes of several people's oral cavities, hopefully more than 3, and we find their overlap. And that overlap of these healthy individuals is going to be the core healthy oral microbiome. And we want this core microbiome because we use it as a baseline for determining what a healthy patient looks like and what an unhealthy patient looks like. So any deviations from this core oral microbiome can be used as kind of an indicator of something not being right in the microbiome and suggesting some sort of disease that's happening. So if somebody doesn't have this core healthy microbiome, then it's probably due to one of or all of these three factors. So the first is abnormal proportions. As I mentioned at the beginning, the symbiotic relationships between these bacteria in the biofilms is very essential in maintaining the health of those biofilms and the surrounding area. So if any one particular bacteria or any two or three particular bacteria begin to overgrow and overpower the other bacteria, then their byproducts won't be able to be efficiently used by the other bacteria. So those symbiotic relationships will be unequal, and they will, the byproducts will then go on to cause harmful effects elsewhere in the mouth or in the body. Having these abnormal proportions also aggravates possible parasitic and opportunistic relationships to emerge. So the bacteria in our mouth don't really care about the survival of other species of bacteria or the survival of their host. All they want to do is survive themselves and reproduce as much as possible. So if they get any sort of opportunity to overgrow and take advantage of their surrounding environment, then they will. And one way of doing this is by certain nutrients being introduced into the mouth, such as sugar. But moving on. The third factor is virulence factors. Again, these are the things that create that protective layer around the bacterial layer in the biofilm. But if the wrong types of bacteria, or not necessarily wrong, but the ones that are unhealthy to us, if they start releasing virulence factors, then it can be a lot harder for other bacteria to keep them back in check and resume those normal proportions that allow for a healthy oral microbiome to carry on. Things that can aggravate or even cause these three things are poor oral hygiene, so not brushing and flossing your teeth. Immunological disorders, again, with the saliva, the inflammatory responses and the immune responses that can be elicited from bacterial production. And then genetic composition, the main thing there is the saliva production that is possible in someone's mouth. So the disease that we are most familiar with in terms of the oral microbiome is dental caries or cavities. So we're just going to quickly go through how that looks. So first we start out with a nice clean tooth, freshly brushed and flossed. And then we have the natural aggregation of bacteria on that tooth just from like swallowing something or eating something or just licking your teeth. However, if we introduce an influx of a specific nutrient such as sugar for cavities in particular, when we have this influx of a nutrient, we also have an influx in the reproduction of the bacteria that eat that nutrient. And that just happens to be Lactobacillus acidophilus, Streptococcus mutans, and Streptococcus cereus. These are the three that we've identified so far. There's probably, but these three is a clear evidence showing that high amounts of these bacteria eventually indicate or lead to tooth decay. So when sugar is in high enough concentrations, these bacteria reproduce at a much more rapid rate than the other bacteria, and then their byproducts, which happen to be very acidic, are not used by the other bacteria, and they end up creating a very acidic environment on whatever tooth they're growing on, which encourages the leaching of calcium and other minerals from the tooth, and also encourages general decay of the tooth. So over time it leads to tooth decay, a sad tooth. Now, how does the oral microbiome and these diseases that occur there really transfer to the rest of the body? Well, there are a couple different ways that that can happen. The first is via dental caries. So if a dental carie is allowed to go on for too long or long enough, it essentially creates a hole in the tooth where the bacteria can pass through that initial dentin and enamel layer into the center of the tooth and travel to the root, where it can cause a periodontal infection. And this infection will cause inflammation that can then be transmitted through the rest of the body. And the second route is through the periodontal pocket, which is the area between your gum line and your tooth. This is the area that you clean when you floss. And it is also a prime aggregation point for bacteria to grow and thrive because it doesn't get disturbed by any other force besides flossing. And it's like a nice, warm, moist environment for bacteria to grow in. So if too much bacteria aggregates in the periodontal pocket, then it can cause inflamed gingiva or gingivitis. And this inflammation and also the bacteria themselves can leach through the gum line right here and reach the circulatory system and travel through the rest of the body. So, as you can see, inflammation is a huge thing here, and the inflammation that's occurring in the oral cavity can transfer to the rest of the body through the gum line and into the circulatory system. But inflammation doesn't necessarily have to be seen in the entire mouth. You can hit one of these abnormal proportions in any single part of the mouth, because each part of the mouth is going to have their ideal proportion of bacteria. So if there's inflammation occurring in any of these sites in the mouth, then that inflammation can travel through the gums and to the circulatory system and travel through the body, causing inflammation in the body, which causes unnecessary stress on our organs and eventually leads to systemic disease. Now, the first disease I'm going to talk about here is very inflammation-based, so it's very based in this pathway, and it is cardiovascular disease. So as you can imagine, all of this starts with bacterial aggregation in the mouth, specifically Porphyromonas gingivalis and Streptococcus sanctis. These are the two that have been identified in this particular study. Once these two bacteria are present in high enough concentrations in the mouth, they are more likely to leach through gums, enter the circulatory system, where they can attach to fatty plaques and cause blockages, and they can even incite inflammatory responses, which causes the blood vessels to swell and encourages clots to form. So by using this information, what if we took the microbial profile of a patient's oral cavity and we found really high concentrations of these two bacteria, we could hypothesize that that person is at a greater risk of cardiovascular disease and give them the according recommendations of lifestyle changes or even possibly medication to prevent the onset of cardiovascular disease. The second disease I'd like to talk about is Alzheimer's. So Alzheimer's is a disease associated with memory loss and general mental degradation. As you can see here, it's a physical brain degradation that can be seen. Currently, the only two ways to really diagnose Alzheimer's is either by observing behavioral symptoms, so memory loss, or by observing brain tissue. So in the brain tissue taken from deceased Alzheimer's patients, we can see neurofibrillary tangles and amyloid plaques, which are not present in a healthy brain. The problem with these two diagnostic methods is that, first of all, the brain's tissue samples are taken from deceased patients, so treatment really isn't an option there. And once the treatments we have available for Alzheimer's currently are mostly to slow down the progression of Alzheimer's rather than reverse the effects that the disease has already caused. So if the person is already showing symptoms, then they don't have that much time left, most likely, even if we start treatment right away. So having an earlier diagnostic method would be very helpful in this disease. So what this study did in particular is that they analyzed the brain tissue of deceased Alzheimer's patients. And they also analyzed the oral cavities of currently living Alzheimer's patients. And they found that both samples contain the same types of virulence factors. And this, again, virulence factors are the compounds that bacteria release, and they reflect a specific environment that fosters a specific composition of bacteria. So this study, they didn't necessarily identify a specific bacteria that's associated with Alzheimer's, but they did find that there is most likely a specific ratio and a specific composition of bacteria that create an environment that elicits inflammation in the mouth, which travels to the brain and may have some sort of direct correlation with Alzheimer's. So this, again, this could be used by, if we analyzed the oral cavity of a patient and we found this sort of virulence factor environment, then we could hypothesize that they might be at greater risk of Alzheimer's. Moving right along, the next slide is not really as inflammation-based as the past two, but it's still pretty interesting, I think. So this study, basically, they just compared the oral cavities, the oral microbiomes of patients with hepatitis B-induced chronic liver disease and those without that disease. and they found that the diseased patients had a much higher ratio of firmicutes to Bacteroides in their oral microbiomes. Firmicutes include things like Streptococcus and Lactobacillus, like I've mentioned before. And although Bacteroides were found in lower concentrations in the unhealthy patients, it doesn't necessarily mean that all Bacteroides species are healthy for a person. They're actually associated with metabolic disorders when they're found in high concentrations in the gut. So again, it's not the presence of specific bacteria, it's the ratio of the bacteria that ends up causing the problem. And another thing that this study found was that they found a very common theme when they were comparing diseased versus non-diseased patients. In the healthy patients, they found a huge diversity of bacteria, all with pretty equal concentrations, whereas in the unhealthy patients, they found a much less diversity in very abnormal and unequal quantities or ratios. They called this transition dysbiosis. And through all the research I've been doing and putting this presentation together, I found that dysbiosis is really a key identifier for identifying disease in an oral microbiome, because the top one here, the huge diversity and equal ratios, that's what the core oral microbiome is going to look like. And then this dysbiosis is a very strong deviation from that. And then finally, this last one is something I just had to include because, as we say in biology, there's no such thing as absolutes. You can never say never because there's always an exception. This one is about pneumonia. So pneumonia is a disease of the lungs where the alveolar sacs in your lungs fill up with fluid and it makes it hard to breathe and gives you a really nasty cough. And this disease is associated with either a bacterial, fungal, or viral infection by pneumonia. And this, of course, is an organism that wouldn't be present in your mouth or your body already. Once it's present in your body, you would get this disease pretty quickly. But you would still be able to diagnose somebody with pneumonia if you analyze the oral microbial profile of their mouth. Now for pneumonia, it has a pretty fast onset time and it's relatively easy to treat compared to Alzheimer's and cardiovascular disease. But I'm hoping that with these examples I'm giving, I'm really honing in on the point that the oral microbiome can reveal a lot about someone's health and possible diseases that they may have. So let's move on to the implications of all this stuff. Why do we care? So as Pritchard puts it, this offers the tantalizing opportunity that by controlling the oral specific microbiome, clinicians may treat or prevent a range of chronic inflammatory diseases orally. So what he's saying here is basically the first part of the implications is that by using the information we can gather from a person's oral microbiome, it can really help us with early diagnosis. And early diagnosis is very attractive to medical professionals because the earlier you diagnose somebody, the earlier you can treat them, and the greater chances of their recovery. The second part is that by using the specificity of each patient's microbiome, we can better cater our treatment to their specific needs. And this is something called personalized medicine. So if we already have this information about the specific genetic makeup and the specific microbes that are alive in a person's body, we can adjust things like dosages, or if there are specific, if there are several types of medications available, we can confidently choose one in particular that would best assist the recovery of our patient. So those two things really make incorporating the oral microbiome into common dental practice very key. However, before we can really incorporate these findings into dental medicine, there are a few things that we need to accomplish first. The first is having a clear picture of microbial rules in health. So as I mentioned, the core oral microbiome is going to be key in creating that baseline of what a healthy microbiome looks like. Once we find that core oral microbiome, we need to get a clear idea of how each of those groups of microbes really affect our health so that we can understand the inner workings of the microbiome better. And then the second one is microbial characteristics of specific diseases. So like the past four examples I gave, just having the specific associations between diseases and either microbial ratios or the presence of specific microbes. We need to know the exact microbial characteristics of the diseases that we want to diagnose. And then finally, having proper diagnostic methods. So currently you can take some sort of sample, like a swab of spit or like a poke from your periodontal pocket, and send it off and get a pretty basic bacterial profile made for a little less than $100. But once we have more information about what specific microbes and microbial ratios we're looking for, we can hopefully cut that cost down a bit, which would be very beneficial considering dental care is already pretty expensive. So just in case you guys aren't planning on applying all this to your dental practices, I have some recommendations for you guys to foster a healthy oral microbiome. The first is to cut down on your sugar intake. So this is pretty obvious, you guys have been told this all your lives, but hopefully now you kind of understand why a bit more. So once you have a lot of sugar in your mouth, it gives a lot of nutrients to the bacteria that do not like to let your teeth survive. If you have too much sugar, these bacteria are going to overgrow and produce byproducts that will rot your teeth. So reduce your sugar intake. Second is to eat a very diverse and high fiber diet. The diversity again will correlate with the diversity of your microbiome and make it harder for specific bacteria to overpower others because you'll just have a lot of diversity in your mouth and in the rest of your body. And then the fiber is just because the fiber, similar to how sugar encourages the growth of the bacteria that are harmful to us, fiber encourages the growth of bacteria that are good for us. So eating fiber is good. And third, fermented foods. As I mentioned at the beginning of this presentation, fermented foods already contain bacteria that create byproducts that are healthy for us. So this is kind of like a cheating way to incorporate lots of good bacteria without having to eat a lot of plants. I'm not saying you can do this instead of eating plants, but it's a good kickstarter. And then finally, become best friends with your toothbrush and floss, because these biofilms, especially if they have those virulence factor protective layers, there's really no chemical way of removing them. You can't just swish your mouth with mouthwash and call it a day. You really have to get in there and physically remove those biofilms if you want to prevent the overgrowth of bacteria. Wrapping it up, I just have some acknowledgments. So first of all, David Hall, he's my advisor and my biochemistry professor. He's helped me a lot these past 4 years and taught me a lot in biochemistry. Maybe you'll want to major in it, so thank you, Dave. Brian Piasecki, he's my microbiology professor, obviously contributed a lot to this presentation. I think I said microbes like 10,000 times. Deanna Donahue, she's been a great advisor as she's just a very kick-butt woman in science, great role model. I've loved getting to know her. Lawrence University in general, the chemistry departments, biology departments, ceramics department for helping with my stress management. Dr. Donald Curia and Dr. Michael Goldberg, these are both dentists that I've shadowed and interviewed, and they really helped me contextualize this presentation, so thank you to them. Karen McGee, she's my mom. Can't say enough good things about her. Wouldn't be here without her, so thank you Mom for everything. And then my classmates and peers, again, wouldn't be here without them. I probably would have dropped out at this point if they hadn't been here to support me. So yeah, thank you. Thank you, everybody. And then, are there questions?
Have at it. Go for it.
So I know, like you mentioned, and I also know, like through personal interaction, that you've like shadowed dentists. So I was wondering, like, how much you see this topic, like, or how much did you observe, or even like if you heard this sort of topic being discussed, like in practice, and like, do you think that dentists today have a good grip on? I mean, I know you mentioned this is new research, but do you think they have a good grip on like the impact of like your oral microbiome besides just at the level of like your mouth, like cavity causing and things like that? Or what is your take?
Right, yeah. Well, the dentist that I actually shadowed doesn't do this, but Dr. Curia, he's the one who I shadowed. He doesn't involve microbiology in his practice at all, but Dr. Goldberg, who I just interviewed, he incorporates it to an extent. He would like to incorporate it more, but we need to do more research about the specific connections. But he mostly does it to make sure that like when he gets new patients, he always analyzes their microbial profile, especially if they've had orthodontic work done. So if they have like a retainer that they wear, he always checks the microbial profile of it to make sure it's not fostering a harmful ratio of bacteria in their mouth. So, but nothing to do with like systemic disease quite yet.
So, do you have any sense in the work that you're presenting how much they understand the correlative versus the causative effects? Because most of the implications you talked about are taking advantage of just correlation, right? When we see this, we know this. We don't know what's causing and what's affecting. But there were a few things that you said that made me think that maybe some of these scientists are looking at causation, like when you suggested that inflammation can travel from the mouth into the body.
Yeah.
So what do you know about the causation effects?
Sure, yeah. Well, I think particularly for cardiovascular disease, they were really trying to make the argument for causation because they have like specific examples of how the bacteria that originate in your mouth can attach to fatty plaques and stuff in your veins. As for the Alzheimer's, I don't know exactly what the causation versus correlation is, but yeah, at this point, I mean, it's really hard to say. It's new research. I think, I think there's the other problem is that a lot of oral bacteria are just found in the rest of your body in general because there's no, aside from like the differences in pHs and the oxygen availability, there's no reason why they couldn't travel to the rest of your body. So it would involve a lot of like dissecting to figure it out. We're not quite.
So the baseline oral microbiome that was the Venn diagram of all, you had 3 separate groups. Do people fall into different categories if you're going to get the baseline? And you had 3 groups. Is that?
The 3 was just because I thought it was a nice number to show like a good amount of overlap. You would have to take it from like thousands of people to get a good idea of what the real overlap is. As for there being groups, I can't speak to the oral microbiome specifically, but I have read about there being like different types of like gut personalities. So like, well, that's what the but just like there are specific trends in the types of there are trends in groups of people who have like specific ratios of gut microbiomes, and that tends to affect things like their weight and their activity levels and their risk of certain diseases. So I'm sure there's something like that for the oral microbiome.
Great, let's thank the speaker again.